Good morning to all. I'm Daphne, working as nursing tutor in Emmanuel Arasar College of Nursing, Natalam. Today we can see the blood-brain barrier in detail. So we can see the anatomy of blood-brain barrier. So introduction is to maintain the normal brain function, the neuronal environment must be preserved with a narrow homeostatic range. This requires tight regulation of transportation. That means the blood-brain barrier is made up of tightly packed cells, molecules and ions between the blood and the brain. Such tight regulation is maintained by unique anatomical and physiological barrier formed collectively in the central nervous system. So this blood-brain barrier is made up of tightly packed cells. Remember it, okay? Next is the neurovascular unit. The term blood-brain barrier has a long history, but it does not adequately encompass a wide range of morphological features and functional characteristics. For this reason, the term neurovascular unit is being increasingly used. It comprises of PB gate, endothelial cells, pericytes, microglia, astrocytes, basement membrane, tight junction. It consists of three barrier layers. It contributes to the separation of blood and the neuronal tissue. First, it is made up of highly specialized endothelial cells, EC layer, comprising blood-brain barrier and partitioning the blood and brain interstitial fluid. The blood CSF barrier with the choroid plexus epithelium, which secretes the specialized cerebrospinal fluid, and the arachnoid epithelium, separating the blood from the subarachnoid CSF. So BBB blood-brain barrier components include PB gate. It includes endothelial cell layer and basement membrane combined together to form adjoined by tight cell-to-cell junction proteins and pinocytic vesicles. The endothelium is surrounded by pericytes, astroglial food process, form additional continuous stratum that separates blood vessel from the brain tissue. So here the blood-brain barrier, that is BBB, is made up of endothelial cell, basement membrane, as well as astrocytes. Okay, astrocytes are the supporting brain cells. So first is endothelial cell layer lining brain capillaries exhibits greater number and volume of mitochondria, augmenting the selective molecular permeability of BBB. There is selective entry in the blood-brain barrier. The basement membrane encompasses pericytes and endothelial cells and is closely adjacent to plasma membranes of astrocytes. Astrocytes are the supporting brain cells. In feet, enclosing the cerebral capillaries. Transmembrane proteins, junctional adhesion molecule 1, occludin and claudin, and cytoplasmic accessory proteins, sona occludens, zingulin, AF6, 7H6 establish tight junctions between adjacent endothelial cells. So this blood-brain barrier is made up of tightly packed cells. Okay, junctional adhesion molecules maintain tight junction properties, as I said earlier. Claudins facilitate tight barrier capabilities. Occludens and sona occludens regulate targeted signaling. Pericytes are enveloping brain microvessels and capillaries found in close proximity to astrocytes and neurons. Pericytes play a critical role in the formation and maturation of blood-brain barrier. Pericytes control cerebral blood flow by regulation of capillary diameter through actin fibers in the pericytic cell body. What is astrocytes? Astrocytes are the supporting brain cells that interact with pericytes and microvascular endothelial cells by end-feet protrusion ensheathing the capillaries. Interaction may also exist with smooth muscle cells at arterioles. Arteries divided to form arterioles. Astrocytes play important role in maintenance of blood-brain barrier, in homeostasis of extracellular concentration of transmitters, metabolites, ions and water. Also serve as stem cell during development. Interaction between astrocytes and neurons determine synaptic transmission. Thank you.